Good evening. My name is Alexandros Patsilinakos, and I am the head of Computational Chemistry at Sibylla BioTech. First of all, I would like to thank the organizing committee for inviting me to give this talk. Sibylla BioTech is an early-stage drug discovery company. The founders invented an innovative pharmacological platform that enables the discovery of new drugs for untreatable diseases. Indeed, we identify untagged protein degraders that act during protein folding. This means, first, a new class of targets, namely intermediate protein folding states. Second, new mechanism of action consisting in protein degradation by interfering with the folding pathway of the target. Third, that this technology allows us to find binding pockets inaccessible in native states. Therefore, our goal is to widen the range of druggable targets currently estimated to be approximately 15% of the human proteome. In detail, while classical drug discovery focuses on targeting the native state of the protein, we act at the stage of folding pathway. Our approach is named PPI-FIT, that stands for Pharmacological Protein Inactivation by Folding Intermediate Targeting. Our rationale is to design small molecules capable of binding one folding intermediate, thus stabilizing a non-native conformation. The cellular quality control machinery recognizes the improperly folded species and promotes its degradation. This approach is extremely powerful because it does not imply a new type of chemistry and can be applied to any therapeutic area. Indeed, we have applied PPI-FIT in oncology, but the first validation was performed in academia by our founders on the prion protein, that is a neurodegenerative target. This validation was published earlier this year, and you can find the details below. Why can we bring this innovation in pharmacology? This is our key differentiator, the possibility to perform the folding simulation of large proteins that are biologically relevant. This is impossible to achieve with unbiased molecular dynamics simulations in a reasonable time. To reach the native state, the polypeptide needs to explore and navigate an extremely rough and high-dimensional energy landscape, therefore encountering energy barriers that need to be overcome. In doing this, the system performs a series of attempts. The rate of the reaction is exponentially suppressed by the height of the encountered energy barriers. Molecular simulations need to follow the polypeptide in this journey, and the maximum time that can be simulated on Earth is a few milliseconds for small proteins that are less than 100 amino acids. If a protein folds in a larger time, it cannot be simulated with standard molecular dynamics. Instead, we apply a bias force that is active only if the polypeptide takes the wrong path and is inactive when it spontaneously proceeds toward the native state. Therefore, we can avoid the simulation of non-relevant detouring and the redundant sampling of backtracking events. In this way, we overcome the timescale inaccessibility problem. The price to pay here is the need to know the 3D structure of the native states a priori. This is an example of what can be achieved with standard molecular dynamics simulations using the most powerful special purpose supercomputer on Earth, the Anton 2, by D.E. Shaw Research. The table here describes the time needed to sample 10 folding events. For instance, the cellular prion protein, PRP, with an estimated experimental folding time of 1 millisecond, would require approximately 1 year of simulation to sample 10 folding events with D.E. Shaw supercomputer. Conversely, we can reconstruct the folding pathway of the same protein by sampling a larger number of folding events in less than 5 days. Our advantage becomes even more extreme when we're dealing with large low-folding polypeptides, such as the alpha-1 antitrypsin. This protein takes approximately 15 minutes to refold in vitro. Anton 2 supercomputer would require hundreds of thousands of years. Yet, we have simulated it in only 6 weeks. Our computational workflow can be summarized in 5 steps. First, the native 3D structure of the target protein is given as input and is typically retrieved by from the protein data bank. Second, the computational platform enables the prediction of the folding pathways of the target at an atomistic level of detail. Third, the trajectories are analyzed to identify relevant intermediate states appearing along the folding pathways. Fourth, druggable pockets exclusively present on the intermediate states, absent in the native conformation, are detected. And finally, a virtual screening of small molecules is performed to identify boundaries of the intermediate states pocket. Let me show here an example of one of our drug discovery programs highlighting the power of the PPI-Fit platform in identifying protein degraders. Here, our technology was applied on cyclin D1. Cyclin D1 is amplified and overexpressed in several types of cancers. Since its native structure lacks any ideal ligandable pocket, this is considered an undruggable target. Currently, the treatment of CDK-dependent cancers relies on CDK4 and 6 inhibitors. Indeed, the inhibition of CDK pathway induces cycle arrest in the G1 phase. However, similar to other kinase inhibitors, they suffer from selectivity problems. In this slide, I'm showing the results of our best hit compound identified by screening 140 virtual hits using cell-based assays. The virtual hits were selected from a virtual screening of 7 million compounds. The graphs show the effect of our best hit in suppressing the level of cyclin D1 protein in 2 different cancer cell lines whose growth is dependent on cyclin D1. As you can see, in HUH7 cells, we already observe a strong effect at 1.5 hours after treatment with an IC50 between 1 and 3 micromolars. Milder but still remarkably significant effect is observed after 9 hours of treatment in MCF7 cells with an IC50 between 3 and 10 micromolars. Here we show the effect of the small molecule degrader in blocking cell proliferation in the same cell lines compared to the two controls, palbociclib, a CDK4 and 6 inhibitor at concentration of 3 macromolars, and nocodazole, a microtubule polymerization inhibitor. As you can see, our hit compound can block cell proliferation to the same extent as palbociclib in HUH7 cells and only at 3-fold concentration in MCF7 cells. Our drug discovery platform relies on ChemAxon tools in 3 main areas. First, the chemical registration, where compounds are registered with the UPAC name, physicochemical and ADMET properties computed with ChemAxon calculators and predictors. The ligand preparation for the structure-based methods, where multiple 3D ligand models are generated with ChemAxon CLI tools with various ionization, tautomeric, and isomeric states. Protein binding pockets often stabilize these alternate forms, even if their prevalence is low in bulk solutions. Moreover, ChemAxon physicochemical properties calculators are used to encode molecules in feature vectors, thus enabling the development and application of QSR and QSPR models. For instance, for experimental endpoints such as binding affinity, bioavailability, and multi-drug resistant substrates. Let me show here more in detail how ChemAxon software is used in our structure-based virtual screening pipeline. Structure-based virtual screening is a powerful process for drug discovery. In this paradigm, a docking program places small molecule conformations within a protein binding pocket, and the scoring function estimates the binding affinities. Ligand filtering and preparation rely on ChemAxon tools. Ligands' physicochemical properties, calculated with ChemAxon CLI tools, are used to pre-filter libraries to enrich them and limit the number of compounds to dock. One of the most popular criteria is the rule of 5, derived by Lipinski from a retrospective analysis of more than 2,000 drugs. The rule of 3 is a similar filter for fragments, with thresholds adapted to their small size. Both correlate the numbers of hydrogen bonds, molecular weight, and octanol water partition coefficient to oral bioavailability and drug likeness. The fundamental task of docking simulation is the post-prediction. Accurate post-prediction depends on a series of factors. One of the most important is the 3D model of the protein and the ligand. Consequently, the ligand preparation step is crucial for a successful structure-based virtual screening campaign. Chemical compound sources often store compounds in formats that include only atom type and bond formation, for example, in SMILES format. Furthermore, database entries typically describe only one ionization and tautomeric state per molecule, and they lack information about chirality and cis-trans isomerization. Therefore, fundamental preprocessing steps must be performed for appropriate ligand preparation, that is, desalting, tautomers enumeration, ionization states enumeration, stereoisomers enumeration, and ring conformation generations. To this end, multiple molecular models are generated with ChemAxon CLI tools with various ionization, tautomeric, isomeric, and property deconformation states. As previously mentioned, protein binding pockets often stabilize these alternate forms, even if their prevalence is low in bulk solutions. The availability of increasingly larger chemical libraries presents great drug discovery opportunities. The number of acquirable compounds is in the order of billions, an amount that will grow. Despite being far from representing the entire chemical space of potential pharmacologically active molecules, estimated to be in the order of 10 to the 60, these chemical libraries enable the discovery of novel chemical classes that are not present in standard in-stock libraries. Structure-based methods can significantly speed up the drug discovery process, but the hardware requirements for running billions of docking simulations are currently unavailable. In screening campaigns based on docking simulations, identifying the highest scoring molecules in a chemical library requires performing docking simulations for each molecule's variance in the entire library. Screening protocols based on ligand-based techniques, such as QSAR, are faster than structure-based methods, mainly due to the use of protein-independent molecular descriptors, such as physicochemical properties, molecular fingerprints, and graphs. Consequently, we are developing a faster virtual screening protocol that can rapidly and accurately evaluate billions of candidates' ligands, binding modes, and scores by adapting an hybrid ligand and structure-based approach. In this accelerated virtual screening protocol, machine learning algorithms are trained on docking scores of small subsets of the large chemical library. The derived supervised learning models predict the binding scores of the remaining molecules, yielding on each iteration, increasingly higher and rich collection of potential virtual hits. This approach is not limited to docking simulations. It can accelerate any structure-based method, such as computing demanding binding-free energy calculations. Thank you for your attention.